Scientists extracted DNA from a woolly mammoth tooth that had been sitting in Siberian permafrost for 43,000 years, used that sequence to synthesize the functional protein that once carried oxygen through the animal's blood, and placed it alongside hemoglobin from Asian elephants and modern humans in a controlled thermal analysis. The results, published in Nature Genetics in 2010 by a team led by Kevin Campbell of the University of Manitoba, established something no researcher had predicted before seeing the data. At temperatures that compromise oxygen delivery in every large mammal alive today, temperatures that force physiological degradation in modern elephants, reindeer, and musk oxen, the reconstructed mammoth hemoglobin continued releasing oxygen to tissue at full efficiency. The protein carried 3 amino acid substitutions in its chimeric beta-delta globin subunit, not found in any living species, and it arrived at its solution through a pathway that evolution has not taken in any other cold climate lineage on record, before or since. A study published in Biochemistry in 2011 confirmed that this temperature-independent delivery profile represents a functional capability that synthetic blood substitutes have never achieved. Hypothermia-induced surgical procedures, cardiac and neurovascular operations in which patient body temperature is deliberately reduced to protect the brain and heart are currently limited by exactly the hemoglobin behavior that the mammoth had eliminated from its own biology. A 43,000-year-old tooth is now an active design reference in the development of artificial blood for living human patients. The animal whose blood produced that finding began its evolutionary history not in the Arctic, but in tropical Africa. That detail requires a moment. The mammoth genus, Mammothus, originated in warm equatorial Africa approximately 6.7 million years ago without a single cold-adapted gene in its entire genome. Every biological mechanism the woolly mammoth eventually possessed, the specialized hemoglobin, the fat metabolism, the thermal sensing, was built through accumulated genetic change on top of a foundation that started in heat. The genome comparison between woolly mammoths and Asian elephants identified exactly where those changes were concentrated. Gene families governing lipid and fat metabolism, adipose tissue distribution, hair follicle and sebaceous gland development, cold tolerance pathways, and circadian biology. The fat metabolism finding carries specific weight. Fixed genetic variants in the mammoth genome altered the way lipids were stored and distributed, producing the dense subcutaneous fat layer and the characteristic fat deposit above the shoulders that insulated the body core in temperatures that would freeze the surface tissue of any unmodified elephant within hours. The circadian finding carries a different logic. Equatorial day length does not vary. Arctic day length spans from 24 hours of continuous darkness in winter to 24 hours of continuous light in summer, and the mammoth genome carries substitutions in clock genes calibrated for exactly that range. These were not small adjustments. They were a full biological reconstruction of an African animal into something the Arctic had never seen before. But the genome did not stop at revealing what the mammoth became. It also corrected what science had assumed it looked like. DNA identified a fixed substitution in a gene called TRPV3, a thermal sensor expressed in skin and hair follicles that governs both temperature perception and the structural development of hair follicles simultaneously. A single amino acid change in that gene altered both how the mammoth experienced cold and the architecture of its hair growth. One mutation, two biological outcomes resolved in the same step. Analysis of the melanocortin, one receptor gene, which controls coat pigmentation across mammals, then overturned a century of visual reconstruction. Woolly mammoth hair recovered from permafrost had consistently appeared orange or reddish, producing the widely depicted reddish-brown animal of museum halls and documentary film. The DNA showed the dominant allele in mammoth populations was consistent with dark brown coloration. The orange appearance of preserved specimens was a product of chemical change after death, a diagenetic artifact that the entire popular image of the animal had been built on. The reconstruction of what the mammoth looked like was wrong. The reconstruction of when it disappeared was equally wrong, and the actual timeline is more difficult to absorb. Woolly mammoths are not 10,000-year-old animals. A population on Wrangel Island in the Arctic Ocean survived until approximately 3,700 years ago, which is roughly 1700 BCE. The Great Pyramid at Giza was completed around 2056 BCE. There were woolly mammoths alive, while Egyptian civilization was constructing monuments that still stand. And the species continued for nearly 900 years after the pyramid was finished. Not as marginal survivors clinging to existence, this was the same species that had been one of the most broadly distributed large herbivores on Earth for 5 million years, now living contemporaneously with the Bronze Age, with written language, with the civilizations whose records we can still read. The textbook placement of the mammoth in the remote geological past is not imprecision. It is simply wrong. Which raises the only question that matters: if the animal survived that long, what finally ended it? The answer that dominated scholarship for over 100 years attributed the collapse to human hunting pressure. The argument had real force. Mammoths had survived the Eemian interglacial between 130,000 and 115,000 years ago, a warming period comparable to the present, without population collapse. Their subsequent disappearance correlated geographically with the spread of human hunters across every continent they occupied. The case seemed settled. A study published in Nature in October of 2021, led by Professor Eske Willerslev of Cambridge University, analyzed 535 permafrost and lake sediment samples from across the Arctic, each containing ancient environmental DNA from mammoth biological material deposited over 50,000 years. By tracking mammoth population distribution and vegetation composition simultaneously, the analysis established that the extinction window aligned not with peak human hunting, but with a specific ecological transition. As the last ice age ended, rising temperatures produced dramatically increased precipitation. The steppe tundra grassland, one of the most productive grazing ecosystems in Earth's history, stretching across entire continents, was replaced by marshland, lakes, and waterlogged ground that could not support the vegetation density those populations required. The herds contracted faster than the grass could be followed. Human hunters were present, and the analysis confirmed the accelerated regional extinction in some areas by up to 4,000 years. But the mechanism was water. The species that had survived 5 mass extinctions was ended by precipitation destroying its food source. That is what ended the species. What the genome reveals about the last individual still alive is a different and darker chapter entirely. Genome analysis of the Wrangel Island population, published in PLOS Genetics in 2017 by Rebecca Rogers and Montgomery Slatkin of UC Berkeley, compared a mainland mammoth from 45,000 years ago, when the species was at full population density, against a specimen from the island's final group of approximately 300 animals. The island mammoth had accumulated a severe excess of harmful mutations consistent with inbreeding and genetic drift at that scale. Large numbers of olfactory receptor genes showed loss of function mutations, stripping the animal of much of its capacity to detect scent. Urinary protein genes, which govern social recognition and mate selection in mammals, had been disrupted. The genome also revealed that at least one individual had developed a translucent satin coat, a mutation that selective pressure in any healthy mainland population would have eliminated immediately. The last mammoths on Earth were not simply isolated. They were deteriorating. Losing function in the sensory and social systems the species had maintained intact for millions of years, on an island the size of a small country surrounded by an ocean that had replaced the steppe they once occupied on 4 continents. And yet, what was preserved in their DNA and the DNA of earlier specimens is what made the final chapter of the story possible. In July of 2024, a team at Baylor College of Medicine published a paper in the journal Cell that peer reviewers described as a result they had not considered physically achievable before examining the data. The team obtained skin from a 52,000-year-old woolly mammoth, in which freeze-drying had converted the tissue to a chemically stable glass, preserving not just the DNA sequence, but the three-dimensional folding architecture of every chromosome, the spatial structure that determines which genes are active and which are silenced in a living cell. Using an adapted technique called Paleo-Hi-C, the team reconstructed the complete three-dimensional genome. It was the first time the chromosomal architecture of any ancient animal had been recovered from fossil material. The comparison with elephant produced a catalog of hundreds of genes active in mammoths and silent in elephants, and hundreds active in elephants and silent in mammoths, not inferred from sequence variation, but read from the preserved activity state of chromosomes that had last been running in a living cell more than 50,000 years before the analysis was performed. In November of 2025, a separate team published the sequencing of the oldest RNA ever recovered from any organism, extracted from mammoth tissue preserved for nearly 40,000 years, establishing that permafrost was capturing not only the genetic blueprint, but the active transcriptional state of the animal's cells at the moment of death. And in 2025, Colossal Biosciences demonstrated mice carrying active woolly mammoth cold tolerance gene edits, including dense woolly coats, cold-resistant fat metabolism, and altered thermal characteristics, as proof of the engineering pathway toward a living animal carrying the full mammoth cold adaptation package. The animal that emerges from all of this shares almost nothing with the one that filled museum halls for 2 centuries. Not a creature of the geological past, but an African animal that rebuilt its blood chemistry through a pathway evolution used nowhere else, that survived into the era of the pharaohs and kept living for nearly 1,000 years after the pyramids were finished, that spent its final centuries on an isolated Arctic island, losing its sense of smell and growing an unusual translucent coat as its genome quietly came apart. It did not disappear because something stronger replaced it. It disappeared because the grass went underwater, and because 300 animals on an island had run out of room to correct the errors accumulating in their DNA. The biology was not lost with them. It was preserved in frozen teeth and freeze-dried skin, and it is now running in living cells for the first time in nearly 4,000 years.